Welcome back to another video. In this module, we are going to talk about the generation of antigen receptors like immunoglobulin molecules and T cell receptors, and a particular genetic concept known as VDJ recombination, the way that we use the special genetics that we find within lymphocytes like T cells and B cells to generate many unique and diverse possible antigen receptors. So let's jump into VDJ recombination, the first video in a three-part series. So if you've been watching these videos, you may remember this slide from early on where I introduced some key differences between the way that we detect self versus non-self in the innate and adaptive immune systems. So within our innate immune response, in order to detect non-self, in order to know that there is an infection present, we primarily rely on receptors known as pattern recognition receptors, or PRRs, which recognize pathogen-associated molecular patterns, or PAMPs. And a key point that I made there was that pattern recognition receptors are generated, or they are expressed, they are encoded, I should say, by genes that are hard coded into the germline DNA. That is to say, they are regular old-fashioned genes that we inherit from our parents and from ancestors going back and into time immemorial, because in fact, pattern recognition receptors are highly evolutionarily conserved across phyla. The idea is that pattern recognition receptors, they rely on traditional genes that are highly conserved. But because we rely on traditional genes, it means, by definition, there's only so many of them that we can encode. So we rely on a pretty broad but limited set of pattern recognition receptors, each of which recognizes a unique set of PAMPs, but not with the specificity that we see with something like an antigen receptor. So in the adaptive immune system, rather than relying on receptors that detect non-self through traditional genetic means, in the adaptive immune system, and this is unique to vertebrates, we've evolved a mechanism by which we can recombine the DNA in somatic cells, and particularly B cells and T cells. These are somatic cells, meaning that they're not gametes. Maybe you've heard of before of recombination happening during meiosis, during the generation of reproductive cells. This is not that. This is highly unusual in that we don't normally recombine the DNA in somatic, non-reproductive cells, but in B cells and T cells we do. And so a process of somatic recombination facilitates the generation of many, many, many orders of magnitudes of unique possible antigen receptors. In B cells, those are going to be immunoglobulins, and in T cells, that will be, of course, the T cell receptor. And the strength or the benefit of this is that this allows us to generate many more unique antigen receptors than we ever would have been able to if each antigen receptor needed its own gene, if each antigen receptor came from some sort of monogenetic encoding. One reason for that, as we'll see, is that the number of unique antigens that are possible that we might need to recognize greatly exceeds the number of genes that we actually find in the human genome. And so if for every antigen that we encountered we had a single gene that encoded a receptor for it, there would basically be no room left over in our genomes for any other type of molecule, any other type of protein. And so we've had to evolve a strategy whereby a more space-efficient sort of method is used, where we actually take gene segments, small gene segments, and we recombine them in many, many unique different orders, essentially, and that gives us a unique combination of receptor outcomes. And we'll look at that process in a lot more detail in just a second. But I just want to highlight that this is something unique to the adaptive immune system. It's something that only happens in B cells and T cells, and only, again, in vertebrates. So a relatively recent evolutionary innovation, which has been very useful to us as we have co-evolved with pathogens. So adding some numbers to the concepts that I went over in this slide, some things I'll bring to your attention is that it is predicted that there are around 10 to the 13th unique immunoglobulins that are possible, which is a kind of staggering number, if you can imagine. So theoretically, we could recognize with our antibodies or with our B cell receptors up to 10 to the 13th unique possible antigens. And this number actually exceeds the number of B cells in the average human body. And so we don't even make enough B cells to account for all the possible antigen specificities that might be represented. There's several reasons for that. One is that out of this 10 to the 13th, many of them are not things that we would actually want. Maybe they recognize self molecules, which is a big chunk of this 10 to the 13th possibility. So we want to get rid of those. We don't want autoreactive B cells. And also, some of the conformations just don't work in terms of the way the amino acids fold. But still, it's helpful to us that we start from such a high place of being able to generate 10 to the 13th unique possibilities. That gives us broad coverage against almost any type of molecular shape that might be generated by a pathogen that we might want to recognize and mount some kind of antibody response against. For T cells, that number is even higher, believe it or not. It's predicted that we can generate up to 10 to the 15th unique T cell receptors, and some estimates are even higher than this. And again, this number is higher than the rough estimation of the number of T cells in the human body. And as I mentioned on the last slide, if you look at these numbers, they are staggering in comparison to the number of known coding genes that are in the human genome, which is only about 200,000. So how is it that with a genome with only about 200,000 genes, can we possibly encode 10 to the 13th or 10 to the 15th unique antigen receptors, which under normal circumstances we might expect to each have their own gene, which is the case for most proteins that we learn about in other biological systems. So what we'll talk about in this video is a special genetic process which sort of allows us within the space confines of the human genome to be able to generate so many proteins with a unique shape, a unique molecular complementarity to individual antigens. So I'm going to start with a slide that really oversimplifies what goes on here, just to highlight some of the key conceptual means by which we make recombined antigen receptors. So antigen receptors are generated through a process called somatic recombination, again, meaning recombination of the germline DNA in somatic cells, B cells and T cells in our case. And it looks something like this. You can imagine that maybe there are some kind of protein that we want to put together, but we want to make a lot of different combinations, and we don't want to spend too much room, too much real estate in our genome to do so. One strategy would be to, instead of encoding each receptor with its own gene, is to actually express a set of gene segments, each of which are by themselves kind of useless. The gene segments do not encode anything useful on their own. But in pairs, if we put one segment with another segment, that together will form a functional exon that can encode the receptor that we want to express. And so in this example, I've given us 6 gene segments that can be recombined pairwise in different ways. And so the way somatic recombination is going to work is that for example, if we want to recombine orange and yellow segments to get a final gene product that is made up of both, what we need to do is actually to bring those 2 gene segments into physical apposition. And we want to bring them next to each other in the DNA. The only way to do that is basically to loop out all of the intervening DNA containing those other gene segments and cut it out and remove it from the intervening, or remove all the intervening sequences. And then eventually, through the process of DNA repair, sort of fuse the 2 segments that we want to keep back together. And so you can see here that leaves us basically with 2 pieces of DNA, one small piece of DNA that contains the genetic material that we've removed, and then the original DNA strand, which now has the 2 segments fused together, which in our example are going to encode a functional gene from 2 gene segments. And so to give you an idea of how this process facilitates going from a small number of gene segments to a larger number of possible recombined genes, you can see on this figure the possible combinations from this setup that I've given you with these 6 different colors gene segments. So if we start with orange and we look at, okay, we can take orange and we can recombine it with green, and then separately we could recombine it with purple and so on, you start to see all these unique gene products starting to add up. You have orange-green, orange-purple, orange-blue, and so on. But we could also start a theoretical gene with any of these colors. We could start it with green, we could start it with purple. The only limitation here is that recombination can only happen in one direction. Remember that DNA is only transcribed in the 5' to 3' direction traditionally. In all of these combinations, you'll notice that we can start with any of the segments, but we can only put a segment, it can only recombine with a segment that is downstream of itself. So you can see that we never, for example, recombine green back to orange. Green can never be in front of orange because orange is upstream of green in this original DNA. So with that rule in mind, how many different combinations can we get if we can sort of only go down, downstream in a downstream direction in terms of the recombination? Well, if you add up all the possible combinations, you can see that even starting with only 6 gene segments, if we apply the rules of somatic recombination, we can actually form 15 unique gene products. So we've more than doubled the number of possible outcomes. And so instead of taking up all the room that 15 genes would take up, we actually have 6 small gene segments. You can see how if you multiply this kind of logic, the efficiency built into this type of system over many orders of magnitude, you can actually get up to very large numbers, in fact, through the process of recombination. So 6 to 15 is cool, but it's not nearly enough efficiency compared to the numbers like 10 to the 13th and 10 to the 15th that we need to generate in order to fully understand antigen receptor diversity. But hopefully this at least illustrates the basic logic behind somatic recombination. Okay, so having gone over that logic, we can now look at the way it actually works in practice. And for this video, we're going to start by focusing on somatic recombination in the immunoglobulin loci. That is to say, how do we put together an immunoglobulin molecule, a BCR, or an antibody? So you may recall that immunoglobulins are made up of both heavy and light chains. In this case, we're going to start with looking at how do we form a light chain, and specifically, how do we form the variable region of a light chain, which here you can see is shown in orange and yellow. Um, the recombination events that we're about to cover are all about forming the variable region of the immunoglobulin because that, remember, is the area that binds to the antigen. It is what needs to vary from immunoglobulin molecule to the next immunoglobulin molecule. We'll see here that there are also gene segments associated with the constant region of the light chain, but they are not recombined. They are not really the subject of the lecture today because they are more structural in nature and there's only a handful of them. So focusing on how do we put together the variable portion of the light chains. Well, as it turns out, there are actually 2 forms of light chain that we encode within our genomes. They are functionally equivalent to each other. There's really no difference in terms of the shapes that they make or the way that they interact with antigen. We basically just have sort of 2 sets of them, and any given immunoglobulin will have a light chain that comes from one set or comes from the other set. And those sets come from distinct chromosomes within the human genome. So you can see that we have what we call the lambda light chain locus on chromosome 22. And so about half of antibodies use a light chain that is derived from genetic material that is encoded in the lambda locus on chromosome 22. And about the other half come from a separate locus that we call the kappa locus, which is encoded on chromosome 2. For now, let's just assume that it's a bit random and that any given antibody, immunoglobulin, its light chain will come from one or the other, sort of again stochastically randomly. The rules for recombination in both, though, are essentially similar. To put together the variable portion of a light chain in either locus, we need to put together a V segment, which stands for, again, variable, or a J segment, which stands for joining. And it's the combination of a V segment with a J segment, just like we saw in the last slide. When we put any of those 2 colors together, that gave us a gene product that encoded for a protein. When we combine a V segment and a J segment in either of the light chain loci, that will give us a functional exon that encodes the variable region of the light chain. And notice that the way this is drawn, that most of the variable light chain is encoded by the V segment, that's represented by most of the structure here being in red. But the J segment is also going to encode an important piece, and especially it's going to encode an N-terminal piece, meaning it's going to be important for putting together the final shape that interacts with antigen. Okay, so how many of these segments do we have? Well, it's different depending on the locus that we're in, but most humans in the lambda light chain locus have about 30 V segments, and they typically have around 4 J segments. And we can recombine essentially any of these V segments with any of these J segments. And so you can already start to think about all of the combinatorial possibilities. And in fact, the math here is pretty simple. If you can combine any one of a set of 30 with any one of a set of 4 to figure out all the combinatorial possibilities, you simply multiply those numbers together. So that means that in the light chain locus, we can start out with already 120, 30 times 4, unique combinations of V and J segments. So now we've gone from 30 plus 4, that's 34 original segments, which can result in 120 unique combinations. And so you can see that these numbers are already starting to add up, although of course we still have a long way to go. In the kappa locus, we can follow a similar logic. Most humans have around 38 V segments in the kappa locus and about 5 J segments. And again, you could multiply 38 times 5 to get the number of combinatorial possibilities here. So now we're up in the hundreds, right, of possible unique light chains that we could generate from a much smaller number of V and J segments. So this is how we put together a light chain for an immunoglobulin. And let's think about the actual process of recombination. What does it look like? Well, in the light chain locus, again, here we can, depending on which locus we're in, either kappa or lambda, we're going to have between 30 and 38 V segments. And again, between 1 and 5 J segments, depending on the person and depending on the locus involved. But the cell is going to randomly, stochastically, ultimately choose one V segment out of whatever number it has and one J segment out of whatever number it has and bring that one V and that one J together and recombine them. And to do so, it's going to cut out all the intervening DNA, remember. So all the DNA in between is going to be removed so that we can physically bring those two segments together. And those two segments together will now constitute a functional exon from which we can transcribe RNA. And then notice that you also have a segment associated with the constant region of the light chain as well. It is there and it needs to be transcribed, but it is not recombined in any way because it does not interact with antigen. So notice again also that the slide is highlighting where different events happen either in the germline DNA or at the RNA or the protein level. So a point I want to make is that the recombination of a V segment with a J segment is a permanent change in the genome of the B cell. Because you're cutting out DNA, that cannot be reversed. The cell only has one copy of DNA. So whenever it does recombine its V and its J segment, that is permanent, and that will encode the light chain that that B cell will make forever, for all eternity, for as long as the B cell lives after that. And so somatic recombination is permanent. However, that recombination event is happening in the DNA, and we can transcribe many copies of RNA off of that recombined DNA exon. And so you can see here that we can make primary RNA transcripts that contain the information encoded by those V and those J segments. So this represents the exonic sequence here, and notice that there are also introns represented by the thin lines, which eventually are spliced out, and we get a final processed mRNA that contains the V and the J, which make up the variable portion of the immunoglobulin. And as you can see, these genetic portions end up encoding the piece of the final protein that are localized about this way on the final immunoglobulin molecule. The majority of this region is made up of amino acids derived from the original V segment, whereas a portion comes from that J or joining segment. And then, of course, the exon associated with the constant region is going to encode the constant region of the final protein as well. Okay, so that is how recombination looks in a nutshell within the light chain loci, lambda and kappa. So let's look at some slight differences in the recombination of gene segments in the heavy chain locus. Because remember, we need not just a light chain, we also need to make heavy chain to make a full functional immunoglobulin molecule. So in the heavy chain locus, which in humans is found on chromosome 14, it looks similar, although we have another segment that we're going to use called a D segment, D standing for diversity because it confers additional diversity in the possible outcomes of antigen specificities that we can generate. And in the heavy chain locus, again, most people have around 40 possible V segments, somewhere around 23 possible D segments, and then up to 6 unique J segments. And we can recombine V, D, and J to form a final, the variable region of the heavy chain. And remember, the heavy chain has multiple subunits. But the variable heavy region is the most N-terminal part of the heavy chain, the part that's going to interact with antigen, which is why it needs to be variable. And then, of course, there are, 3 or depending on the isotype, even more constant heavy segments. And they are also encoded by additional exons downstream. But again, those exons are not generated through VDJ recombination. We're only talking about right now, how do we make the variable heavy region of the immunoglobulin? And you can see here again that most of that final part of the protein is going to be encoded by the V segment, but the J segments and the D segments are also going to have a part to play in putting together the N-terminus of the variable heavy region, which again is key because that is the part that physically interacts with antigen. So let's look a little bit at how the process of recombination looks slightly different now that we have 3 different types of segments to put together as opposed to only 2. So if we zoom into the heavy chain locus, we're in the germline DNA. Remember that we have roughly these numbers of V, D, and J segments, but the cell is going to again randomly choose one D and one J. And one point here to make is that D and J always recombine first. There are various mechanisms in place to make sure that happens. So the cell starts with one random D and one random J, and it brings them together. And recall that that means that all of the intervening DNA between whichever D and whichever J is chosen is cut out. And we'll look at molecular mechanisms for how that happens in the next video. Once we form a functional DJ pair, we need to bring it into proximity now with whichever random V that we've chosen. And again, the DNA is cut out and we bring it together to form a final functional exon made up of all 3 V, D, and J segments. And this exon is what will encode the variable heavy portion of the final protein, the final immunoglobulin molecule. And you can see here that immediately downstream, there's a set of exons also that encode the various constant heavy regions, including the hinge region, which is shown here in this kind of lighter blue color. There are also amino acids that we use in that part of the gene structure to encode that as well. But again, that process is not involving recombination. These are conserved exons that are inherited directly from the germline and are not changed through the process of recombination. Okay, so now we've formed a functional heavy chain, and in combination with whichever recombined light chain the B cell has made, we'll now put whatever that light chain is together with whatever that heavy chain is to form one unique final immunoglobulin molecule that will recognize one unique antigen. And another point to make here is because this is a permanent change to the germline DNA, that means that is the antigen that B cell will recognize forever. But also, because this happens in the DNA, that means that all RNA is coming off of the same template. The consequence or the relevance of that is that all of the immunoglobulins made by a B cell will then recognize the same antigen. A B cell cannot recognize multiple different antigens. It's not making multiple different BCRs on its surface or secreting different antibodies. The antigen specificity will always be the same because it's all templating off of one unique source, which is the recombined VDJ segments within its germline DNA. Okay, so that was the process of recombination. Now let's think about, given the numbers of all these segments, how are we doing in terms of generating all of the possible unique possibilities that I said that B cells are capable of, something like 10 to the 13th? How are all these numbers adding up in order to generate all that possible diversity? Well, to sort of figure that question out, we can look at these numbers in a way that I think sort of help us to imagine how the math will start to work. Remember that we need both a light chain and a heavy chain to make an immunoglobulin molecule. and that for the light chain, that we are going to use V, D, and J, or sorry, in the light chain, we're going to use V or J segments that come from either the kappa or the lambda locus, and that within either of these loci, we have a certain number of variable segments. Again, this varies depending on the individual person, and a certain number of J segments, and that to sort of calculate how many unique light chains we might make, we're just going to do some multiplication. And then similarly in the heavy chain, we can multiply all the possible V segments by all the possible D segments by all the possible J segments in order to figure out the total number of outcomes that are possible, which would represent the total number of unique antigens that we might recognize. So in order to do this math, let's take an example individual. This is one human being who has the following numbers of segments in each of these loci. So in the kappa loci, this person has 35 V segments and 5 J segments. In the lambda locus, they have 30 V, 5 J, and then in the heavy chain locus, they have this number of segments within for each type, V, D, and J. So to calculate the number of possible kappa light chains, that is light chains that are encoded by the kappa locus, we simply take 35 times 5, the number of V segments times the number of J segments, and that gives us about 175. If we do similar math using the numbers of segments in the lambda light chain locus, we get about 150 unique possible lambda light chains or variable regions of the lambda light chain. The number of possible heavy chains or unique heavy chains that come out are simply those three numbers multiplied by each other, as I said, which comes out to about 6,210. And so you can actually see that the introduction of those D segments actually increased the number of possible heavy chains actually by a whole order of magnitude. And so these numbers actually are adding up. They're escalating quite quickly. But this is all the possible numbers of chains that we can make, but that's actually not that helpful. What we need to know is how many ways can we put these chains together in order to actually make whatever number of unique immunoglobulin final proteins, because that is actually what are going to recognize the antigens that we might encounter. So how do we sort of calculate the total possible final outcomes using these numbers? Well, one thing to keep in mind is that immunoglobulins either have a variable light kappa or a variable light lambda, and that is an either or choice. And so the numbers of kappa light chains do not influence the number of variable lambda light chains in terms of the way that they contribute to the overall diversity of what we call the repertoire, or the sort of total set of possible end outcomes. So given that all immunoglobulins have either a lambda or a kappa light chain, what we need to do is figure out how many unique kappa immunoglobulins can we make and how many unique lambda immunoglobulins can we make separately or independently of each other. So to do that, to figure out how all of the possible kappa immunoglobulin molecules, we take the number of possible kappa light chains and multiply that by the number of possible heavy chains. And we get a number around 1 million, as you can see. Similarly, we would take the number of possible lambda light chains and multiply that by the number of possible heavy chains, and we also get a number that's slightly less, but also again around 1 million in terms of its order of magnitude. And then to calculate the total repertoire, we would add these two numbers together. We do not multiply them because they are not being combined with each other in any way. We simply have one set that comes from the kappa locus and one set that comes from the lambda locus, and we'll only get one out of either of those sets. So all of the total possibilities then come to about 2 million, and we think of this number of 2 million as the total combinatorial diversity, the total combinatorial possibilities of the way V, D, and J segments can be recombined to make unique immunoglobulin molecules. And so a key point here is that we went from an initial set of 150, around 150 gene segments, small gene segments, VD and J segments, and all of that recombination, or all the possible recombinations of those 150 segments, gave us 2 million outcomes. Again, this number is already higher than the number of genes in the human genome. So you can see that we added quite a bit of possible diversity without taking very much space in the genome. So you can sort of see the power here of combinatorial diversity to increase our repertoire without taking up too much space in the genome. But of course, you may be thinking to yourself, okay, 2 million is cool, but that only represents 10 to the 6th. And I said that there were 10 to the 13th possible immunoglobulins. So as we'll see in the next lecture, there are additional mechanisms that give us that additional diversity. How do we get from 10 to the 6th to 10 to the 13th? But you'll have to stay tuned for that. For now, let's summarize this video, the first part of our discussion of VDJ recombination. We saw that immunoglobulin variable domains or regions are encoded by discrete gene segments. Light chains are encoded by V and J segments, whereas heavy chains are a combination of V, D, and J segments. In any case, these segments are recombined together to make unique combinations. And in the light chain, we simply put V and J together before ultimately adding our constant region during RNA splicing. Whereas in the heavy chain, we first recombine D and J, and then we add that to V to form a functional exon that encodes the variable region, and then the constant region is spliced on as well. And we saw that we can take around 150 initial gene segments and recombine them in various ways to end up with about 2 times 10 to the 6, about 2 million unique combinations. And this logic, or this, this sort of outcome, is what we call combinatorial diversity of the immunoglobulin repertoire, of the repertoire of all the possible antigens that we might recognize with an immunoglobulin molecule. But as I said, we need to get up to 10 to the 13th because that is the predicted total diversity of the repertoire. So we'll need to see what additional mechanisms exist beyond combinatorial diversity, and we'll take a stab at that in the next video. See you there.